Welcome to the deep dive. When you think about life, really think about it at the molecular level, it's just incredible. Billions of molecules buzzing around, finding exactly the right partner, doing a specific job, and then letting go, all with this amazing precision. How does that even happen? How do they manage this whole molecular dance without always forming those really strong permanent chemical bonds we learned about? Okay, let's unpack this. Today we're diving into, well, the secret handshake of the molecular world. Non-covalent attractions. These aren't the big flashy chemical bonds, the really permanent ones, but they are absolutely essential. For everything that happens in your body. Everything.
Absolutely. And our mission today really is to give you a kind of shortcut to understanding the four main types of these non-covalent attractions. We'll look at what makes each one unique, their strengths, their weaknesses, especially in context, and most importantly, how they all work together in concert to bring molecules together so specifically inside our cells. The goal is simple. You walk away with a clearer picture of these fundamental forces and, why they matter quite so much.
Great. So where do we start this journey? Maybe with something that sounds strong on paper. Ionic bonds. I think most people remember these from high school chemistry. It's all about electron transfer, right? One atom gives, one atom takes.
Exactly. One atom giving up an electron, becoming positive cation, and the other atom gains it, becomes negative an anion. And because opposites attract strongly, they form this ionic bond, creates what we call salt, basically, an ionic compound. And what's really interesting is, yes, they are a type of electrostatic attraction. That pull between the full positive and negative charge, it's powerful. In a vacuum, incredibly strong. Okay, but that's where it gets tricky, isn't it? The in a vacuum part. Because for something that sounds so strong, there's this big catch when you put it into, into a biological system. It seems almost backwards that something so strong can be easily broken apart.
It does seem counterintuitive, but it's absolutely critical for biology. Think about it. Cells are mostly water, right? Around 70%. And water is a very, very polar solvent.
Meaning it has those slightly positive and negative ends.
Precisely. The oxygen side is a bit negative, the hydrogen sides are a bit positive, and these partial charges are fantastic at essentially surrounding and pulling apart ions in a salt. So inside the watery environment of a cell, most of those strong ionic bonds, they're actually dissolved. The ions float freely.
Which is actually useful then.
Oh, incredibly useful. It allows ions to move, conduct signals like nerve impulses, help maintain the right pH balance, tons of things. It's really only in very specific spots, tiny pockets, maybe deep inside a protein where water is excluded, that an ionic bond can exert its full strength, maybe to hold something temporarily in place. Okay, that makes sense. Strength depends on the context. So, if ionic bonds are often neutralized by water, what other non-covalent forces are doing the heavy lifting, orchestrating all this? Let's talk about hydrogen bonds. They seem to pop up everywhere.
They really do. Hydrogen bonds are fundamental in biology, and they're different from ionic bonds. It's still electrostatic attraction, but it's much weaker. We're dealing with partial charges now, not full ones.
How does that work?
Well, you have a hydrogen atom that's already bonded to something very electronegative, like an oxygen or a nitrogen. Think water, H2O. That oxygen pulls the electrons close, so the hydrogen becomes slightly positive. The slightly positive hydrogen is then attracted to another nearby electronegative atom, maybe the oxygen on another water molecule.
Okay, so it's not a full charge attraction, more like a weak magnetic pull between slightly charged parts.
Exactly, like a fleeting attraction. Individually, they are quite weak, much weaker than a covalent bond or even a full ionic bond in a non-water environment.
But they seem so important.
They are, because it's all about the numbers. Their sheer quantity and the precise way they have to line up, geometrically, makes them incredibly powerful collectively. Think about DNA, the double helix. It's held together by millions of hydrogen bonds between the base pairs. They act like the zipper,
right,
for proteins. Those alpha helices and beta sheets, the basic folding patterns, they're stabilized by specific patterns of hydrogen bonds. And water itself, its high boiling point, its ability to dissolve things, that's all down to the huge network of hydrogen bonds between water molecules. They're constantly forming and breaking, which gives water its unique fluid yet cohesive properties.
That constant forming and breaking, that sounds key for flexibility too.
Absolutely. Stability and flexibility.
Okay, fascinating. Now, let's go even more subtle. Maybe the interactions people find a bit harder to grasp, like hydrophobic interactions. You see this with oil and vinegar, right? They just don't mix. What's actually going on there at the molecular level?
Yeah, that's a great visual. And it's a common point of confusion because hydrophobic interaction sounds like the oil molecules are attracting each other. But it's not really an attraction in that sense. It's actually driven more by water.
By water. How so?
Well, water molecules love bonding with each other through those hydrogen bonds we just talked about. They form a very ordered dynamic network. When
Yeah.
you introduce something nonpolar, like oil or maybe the nonpolar parts of a protein, into water, it disrupts that network. Water molecules have to arrange themselves around the nonpolar molecule in a very ordered, cage-like way.
Okay.
And forming this ordered cage is entropically unfavorable. It reduces the randomness, the disorder of the water molecules, which systems tend to resist.
So water kind of pushes the nonpolar things together
I
to
have
minimize
this-
the disruption.
Exactly. That's the key insight. By forcing the nonpolar molecules or regions to clump together, water minimizes the surface area it has to form those ordered cages around. This minimizes the unfavorable entropy cost for the water. So it's an indirect effect, but it's a major driving force for how cell membranes assemble, how proteins fold up correctly with their oily bits hidden inside. Water's dislike for nonpolar things structures much of biology.
Wow. Okay. So it's water being picky, essentially.
You put it that way.
All right. From water being picky, let's shift to the universal but maybe weakest interaction. Van der Waals attractions.
Yeah.
These sound almost negligible.
They are individually negligible almost, but they are absolutely crucial, and they happen between any two atoms that get close enough.
Any two atoms, even if they're not polar or charged?
Yes. Even in a completely nonpolar molecule, the electrons are constantly whizzing around. Just by chance, at any given instant, the electrons might be slightly more on one side than the other. This creates a tiny, fleeting, temporary dipole, a slight separation of charge.
Okay, like a blip of polarity.
Exactly, a blip. And that temporary dipole can then induce a complementary dipole in a neighboring atom or molecule. So for a split second, you get this very weak attraction between the slightly positive side of one and the slightly negative side of the other.
Like microscopic static cling. On, off, on, off.
That's a great analogy. It's incredibly weak individually, like really tiny, fractions of the strength of a hydrogen bond. But, and this is the critical thing, they become incredibly significant when you have a lot of them working together over a large surface area.
The collective power idea again.
Right. Think of a gecko climbing a wall. Its feet aren't sticky in the way tape is. Instead, the microscopic structures on its feet create a huge surface area, allowing for millions upon millions of these tiny, fleeting van der Waals attractions with the wall surface. Individually, each attraction is nothing, but add them all up. The gecko sticks.
That's amazing. So in biology, this allows molecules to...
It allows molecules that have precisely complementary shapes to fit together and interact, like an enzyme and its substrate. The surfaces match closely, allowing lots of these weak van der Waals forces to hold them together just long enough for the reaction to happen. And this relates directly to something called van der Waals radii.
Radii, like the size of the atom.
Sort of its effective size, yeah. It defines the closest distance two non-bonded atoms can approach each other before their electron clouds start to repel. It's like their personal space bubble. For van der Waals attractions to work best, the atoms need to be right at that optimal distance, not too far apart, but not too close either. That perfect fit maximizes the number of weak interactions.
Okay, that makes sense. It's about getting close but not too close over a large area. So, putting it all together now, how do all these different forces, the ionic bonds that depend on context, the hydrogen bonds holding things together, the hydrophobic effect pushing things around, and these numerous van der Waals nudges, how do they all combine to let molecules recognize each other with such incredible specificity? It seems so complex.
It is complex, but it's also beautifully elegant. Molecular recognition boils down to two main things. First, as we just discussed with van der Waals, there needs to be a really high degree of shape complementarity. The molecules have to physically fit together like 3D puzzle pieces, often defined by those van der Waals
The
dimensions.
physical fit, got it.
Second, and this is crucial, as they fit together, there needs to be an alignment of many, many weak non-covalent interactions across the interface where they touch. It's not just one strong bond, but the sum of many hydrogen bonds, maybe some ionic interactions in the right spots, van der Waals forces all over the matching surfaces, and hydrophobic effects positioning things correctly.
So it's the combination of shape and the sum of many weak chemical handshakes.
Precisely.
Now, Our source material mentions something about a two-way arrow being very important here. What does that signify in this context of weak forces and molecular recognition?
Ah, yes, the two arrow. That's fundamental. It represents the fact that these interactions are reversible.
Not permanent.
Exactly. Unlike a strong covalent bond that really locks things together, these non-covalent interactions are designed to be formed and broken relatively easily. Molecules associate, they interact, and then they dissociate. Bind, release. Bind, release.
And why is that reversibility so important?
Because life is dynamic. Processes need to happen quickly, signals need to be turned on and off, enzymes need to find their target, do their job, and then let go of the product so they can do it again. If these interactions were too strong, too permanent, everything would grind to a halt. It wouldn't be a dynamic dance, it would be statues. This flexibility, this constant associating and dissociating enabled by the sum of weak forces, is absolutely essential for regulating everything in the cell, from metabolism to signaling to DNA replication. It allows for rapid responses and fine-tuning.
Okay, let's maybe try and pull this together with a quick summary.
Yeah.
It feels like we've covered a whole spectrum of interactions. At one end, you have covalent bonds. Strong, stable, forming the basic structure of molecules.
Yeah.
Though, as we know, not all covalent bonds are equally strong either, but that's another story. Then we dove into the non-covalent world. We had ionic bonds. Strong in theory, but usually dissolved in water inside cells, powerful mainly in water-free zones.
Right.
Then hydrogen bonds. Weaker individually, using partial charges, but their sheer number makes them vital for structures like DNA and proteins and for water's properties. Then hydrophobic interactions. Not really an attraction, but water pushing non-polar stuff together. Key for membranes and protein folding.
Driven by entropy, yeah.
Or finally, van der Waals attractions. The weakest of all individually, these fleeting attractions between any nearby atoms, but add up powerfully when surfaces match closely, like the gecko's foot. Defined by those van der Waals radii, that personal space.
That's a great summary. And it leads to that really important question, why? Why does nature rely so heavily on these seemingly weak forces for almost all of its complex operations? And the answer, I think, is just elegance, biological elegance. These weak, dynamic, reversible interactions are the key to life's adaptability and responsiveness. They allow things to come together when needed and fall apart just as easily. Think about building molecular machines like ribosomes. They assemble, do their job, maybe disassemble, or signals passing across a cell membrane receptors bind, trigger a response, then release, or proteins folding into incredibly precise shapes guided by the sum of countless tiny interactions. It allows for rapid control, precise timing, and constant adjustment. The van der Waals radii we mentioned aren't just abstract numbers. They define the exact physical spacing needed for this precise molecular recognition to occur effectively. These forces are, essentially, the invisible architects building and rebuilding life, moment by moment, constantly, dynamically right inside you.
It's quite mind-boggling when you frame it like that. So we've really taken a deep dive today into this hidden world of non-covalent attractions, from ionic bonds needing the right environment to the ubiquitous hydrogen bonds, the water-driven hydrophobic effect, and those tiny but numerous van der Waals forces. And we've seen how, together, these individually weak or context-dependent forces create this incredible specificity, this fit, allowing molecules to find each other, interact, and basically run the show.
Exactly. And if we connect this to the bigger picture, I think understanding these interactions really highlights the incredible sophistication of biological systems. Nature has found this perfect balance, hasn't it? Between stability, things need to hold together long enough to function, and flexibility, things need to change and respond. It's this delicate balance enabled by weak forces that allows living organisms to be so dynamic and responsive. It's the power of many small things working together in just the right way. So maybe something to think about as you go about your day. Consider how these seemingly simple, almost invisible forces, when orchestrated just right, how they enable literally everything. Your cells communicating, your muscles moving, even your thoughts forming. It's all underpinned by this molecular dance. What other things in the world, maybe complex systems we see every day, might also be built on similar principles, where countless small, subtle interactions add up to create something incredibly complex and functional?